Hello, everyone. My name is Angela Clark with Propel a Cure for Crohn's disease, and we're here with another episode of our continuing series of discussions with leading researchers in the IBD field. We have with us here today Dr. Alison Simmons. She is one of the senior authors of a study that was recently published in the journal Nature on the formation and behavior of fistulas in Crohn's disease. Dr. Simmons is a clinician scientist who researches intestinal immunity. She's been involved in molecular mapping that has highlighted novel avenues for tackling unmet needs in tissue inflammation. Dr. Simmons trained in medicine and gastroenterology in London, Cambridge, and Oxford, and obtained her PhD in molecular immunology from Oxford University. She is a fellow of the Academy of Medical Sciences, director of the MRC Translational Immune Discovery Unit, and professor of gastroenterology at the University of Oxford. She's an honorary consultant gastroenterologist at the John Radcliffe Hospital. Welcome, Dr. Simmons. We are so pleased to have you with us today to talk about this topic.
Oh, well, thank you for that really kind introduction. I'm very pleased to be here.
Now, as a member of the IBD community, I have to say that fistulas in Crohn's disease have to be one of the most challenging complications of IBD, both from a physical and a mental standpoint. It's a hard subject to talk about sometimes. It's sensitive, and there's just a lot of misunderstanding about it. And we're really eager to hear about your findings and your knowledge of this subject. So thank you for sharing it with us. Can we just start with the beginning, and can you tell us what a fistula is?
Yes, so a fistula is an abnormal tunnel-like connection that forms between two body parts that are not normally connected. And in Crohn's, and fistulas are not exclusive to Crohn's, but they can commonly, as you occur in Crohn's. Ongoing inflammation, basically in the disease over time, often contributes to burrowing through the intestinal tissue and creating a passageway that connects the intestine maybe to the skin or to another bit of the intestine or to the bladder or another organ. So, of course, they are incredibly incapacitating.
Yeah. So I've seen through research that at least 30% of people with Crohn's will develop fistulas at some time. Is that fairly accurate?
Yes, that's correct. It is. Many of them are perianal fistulae. But as I say, fistulae can occur between internal organs and, can range from being an incredible nuisance, causing a degree of morbidity, to something which is really very seriously incapacitating.
And what drew your team to investigate fistulas?
Well, for several reasons, really, largely because they represent one of the most disabling complications of inflammatory bowel disease, and yet, paradoxically, they remain underexplored from a science perspective, underfunded, arguably, and they're poorly integrated into care models. And the first author of the study, Colleen McGregor, who's a gastroenterology trainee, felt very passionately that she wanted to really focus in on this particular complication of Crohn's because of its understudied nature. Fistulae can signal more aggressive Crohn's disease, but treatment, despite that, treatment advances have lagged behind the rest of the condition, I think it's fair to say.
And fistulas are apparently difficult to study. Why is that?
Yeah, I think that's for several reasons, really. One is sort of biological reasons, in that they are incredibly heterogeneous. That is to say, they can occur at many different anatomical locations. So not all fistulae are the same. They differ in location, they differ in tissue depth, they may differ in the kind of anatomy they display, so whether they're a single tunnel or whether they form a like branching tree structure within the tissue, whether they're the degree of inflammation associated with them, the degree of infection associated with them, or abscess or stricture formation linked to them. So they're very, very different. And so that makes it difficult to compile a cohort large enough to take into account of all those differences to make scientifically meaningful sort of deductions. And the second reason is almost certainly technical, in that until recently, very recently, it's not been possible to actually study the rare tissue, the cells that line the fistula tract. So, when you actually take resect a piece of tissue with a fistula tract in it, the number of cells that actually line the tracts are tiny numbers. And so to get biologically meaningful data from very tiny numbers of rare cells has been very challenging. And recently, a number of new technologies have enabled us to really drill down on that information within those rare cell types and actually reveal the pathology in much greater detail. So biological and technical reasons.
So tell us more about the study. Can you give us just an overview of some of the steps your team took through this study?
Yes. So first of all, the first job was to collate a cohort of samples that would allow us to really compare and contrast across all these heterogeneous manifestations of fistulae. So we compiled a cohort of, in the first instance, 68 specimens kindly donated by very generous patients. And we also had some control samples from fistulae from other causes and so forth. And so that allowed us to then conduct a series of high-resolution molecular profiling assays, which allowed us to sort of understand the gene expression coming out of each single cell that made up the tract, but also the spatial location. So we undertook what's called a spatial transcriptomic analysis, which allows us to place the cells in tissue space and understand how they're talking to each other. And then the third thing we did was that we studied the collagen matrix in which all these cells lie, because the cells making up the tract actually contribute to the matrix formation. And we studied that using a sort of advanced imaging computational approach, and we could correlate those changes with the individual cells lining the tract, and that gave us a much more granular vision of what was going on, essentially. Enabled us to reveal kind of hidden biology, if you like.
And one of those great findings was what you found was the presence of something called rogue fibroblasts as part of the formation. And this is, to me, this was very just unbelievable. Can you explain what fibroblasts are and what they normally do and what makes these types so different in fistula?
Yes, so fibroblasts are cells which kind of, could be thought of as general support cells. They form the sort of, tissue. They make up the tissue, the substrate of the tissue, if you like, and their main role in the gut, they come in several flavors, but they're one of the key hallmarks of a fibroblast in general is its ability to secrete matrix and make secrete collagens, which create the pattern of the tissue and support the specialized bits of the tissue like the surface layer, the epithelia, and the lymphoid structures, the immune system within the gut tissue. So they're cells which make up the sort of anatomical backbone of the gut, and they secrete matrix to allow the shape of the gut to be maintained. And they're very important in wound healing. And what's really happening in Crohn's is that you're getting these very nasty depulses which do not heal properly. And so it turned out that when we surveyed these fistulae using the techniques that I mentioned, we detected a series of rogue behaviors in these fibroblasts lining the tracts, which allowed them to sort of burrow, if you like, through the tissue and behave like abnormal wound healing cells rather than perhaps scar cells, which you'd expect in a normal healing response. They also interacted with immune cells in a way which formed a sort of self-sustaining circuit or niche to drive this abnormal wound healing response and perpetuate the fistula tract formation. And so I think this is probably not very well described, but essentially what we found in the study was that fistulas are not just sort of holes; they're organized living structures lined by these FAS cells, rogue fibroblasts, which are enabling tunneling and also laying down an abnormal matrix to stabilize the tract. So all in all, a thoroughly abnormal wound healing response.
And this was really a groundbreaking discovery, correct? This had not been known before.
This has not been known before, simply because, as I say, it was difficult to kind of survey the behaviors of these cells in a holistic fashion. And I think these new technologies are allowing us to really kind of do this for the first time. Yes.
And one interesting thing that jumped out in the study too was that this abnormal behavior of these rogue fibroblasts are actually how they behave before birth. Is that correct in the developmental stage?
Well, what we found was in order to, so this is what you're alluding to, is the programs that they switched on to allow them to misbehave, if you like. And they, like many abnormal wound healing responses across the body, what they had done is switched on developmental programs that you normally see in the fetus to drive normal patterning, normal gut formation, but they kind of switched them on aberrantly. And in the context of this inflamed milieu, this abnormal expression is indicative of, ending up with a pathology like a fistula, essentially.
What other findings in your study do you consider the most exciting about fistula development and behavior?
Well, I think we found that different fibroblast zones perform different jobs. So in the fistula tract, some of the fibroblasts drive inflammation, while others remodel collagen, and others still form rigid fibrotic walls, stabilizing the fistula tunnel. I think that was quite interesting. And the other feature was that when we surveyed deep ulcers present in Crohn's patients, there were some fibroblasts right at the bottom of those ulcers which had properties which were similar to those that the FAS cells, the rogue fibroblasts, and it's sort of giving an indication that perhaps these are precursor cells in some way, something to be further worked on.
Some patients have other complications, like strictures, for example. How have you seen these other complications interact with or potentially even cause fistulas? Can you tell us something about that?
Well, I mean, that wasn't, strictly that wasn't something that we studied within this study, but in general, I mean, of course, there is an interrelationship there. And, strictures in themselves raise mechanical and microbial stress that can promote fistula formation, while fistulas and abscesses worsen scarring, stricturing, and narrowing. So this is probably why some patients with one complication are at risk for the other, and why, of course, early aggressive control of inflammation is critical to prevent both of those things. Yes. Yeah.
And what insights from your study did you find that may help us in the future prevent fistula potentially?
I think at a very high level, I think what our study kind of emphasized was that fistulas are not just simply inflammatory complications. They are structurally engineered wounds maintained by abnormal fibroblasts and immune signaling, and treating them will likely require new therapies that target wound healing biology and tissue remodeling, not just inflammation. And the armory of treatments that we have for IBD at the moment really focusing on inflammation. I think using the spatial approaches, which will become used, much more commonly across all sorts of contexts in IBD, these spatial approaches really highlight different bits of anatomy or pathology that you could go in on when thinking about designing new treatments. So I think, there's a lot of opportunity there.
And can you tell us more about that? What has been the standard of treatment to this point for fistulas, and and can you further expand on what therapeutics might look like for their treatment?
Yeah. So, I mean, I think it's fair to say that current fistula-based care is often reactive. So there's no current method really to predict whether you're going to form a fistula or prevent fistula formation. And that it's anatomy based. So, for example, medicines or surgery or combination of both will be used depending on the anatomy of the lesion to actually treat the patient. And as I said before, the kind of, all the therapies that we have are really inflammation centered. So they aim to suppress different aspects of inflammation and the immune response in fairly non-specific ways, often not taking into account the underlying cellular pathology at such a granular level. And I think in the future, we'll move forward to a point where, fistula-based care may be predictive. We might get to a point where we can prevent fistulae by being able to predict when they're going to form, using molecular phenotyping. And that when we think about fistulae, we'll be thinking more along the lines of wound biology centric approaches to sort of promote wound healing and intestinal regeneration, healthy regeneration, and immune restoration, as opposed to simply suppressing inflammation. I think that's where things will go.
So at this point, I suppose some biologics might help fistulas, but not uniformly, I understand. So you foresee these therapeutics as perhaps separate therapies.
New therapies, absolutely. New therapies is what we need to really promote healing and prevent them occurring in the first place. And I think, there's a huge amount of activity in the whole field. And I think there's a load of opportunity there to really go in on this at this point in time. Yeah.
As part of this study, you created one of the largest data sets of this type and have made it publicly available. Can you tell us more about that and how you hope other scientists will use that?
Well, I think those of us in the field utilizing these very large scale approaches to generate big data, really want to see the data used, and the data will be used by the community to inform. I mean, there's several key things, particularly with regard to fistulae, is can we make better cellular or tissue models of this disease? Because that will help us screen through potential new treatments, examine pathways that might be used to resolve the pathology. So we need new models. And, a data set like this, like many others, acts as a great catalyst to, start investigating novel biological pathways that might be driving disease, so that it's an unbiased screen which throws up a load of possibilities as to what might be going on, and so people will use this as a way to inform further biological studies. And the other important approach, which is used where the data will be used, is for AI-based modelling of causal molecular drivers, and this is an important way often or one method to inform new drug discovery, essentially.
Are there any misconceptions about fistulas that you wish patients or even clinicians better understood?
Well, I think probably maybe from a patient perspective, fistulas, people think fistulas only occur in severe disease. That's not the case. There probably is the surgery alone can cure them. Is a misconception that surface closure equals healing, and perhaps some of those misconceptions could delay accessing effective treatment and maybe worsen outcomes. I think that might be something. Yeah.
That's very interesting, because I know that surgery is sometimes what a lot of times will finally rid a patient of a fistula. So it would be great to have tools beyond that to prevent recurrence. Absolutely. And what unanswered questions remain about fistula formation that you're most eager to explore, perhaps?
Gosh, I think we'd like to know what molecular programs turn ulcers into tunnels. At least in some cases, ulcers are likely to be the starting point. Not all, but at least in some cases. And if we could predict, how that was going to emerge, we'd have better ways to prevent. Why does wound healing fail in fistula tracts? I suppose can we predict penetrating disease before anatomy changes, and can we shift therapy from closure to prevention? I think those are the key things, really. And under that subheader, surely we can now make some better models, in vitro models, that allow us to really focus in better on aspects of the pathology for the design of improved treatments. Yeah.
Dr. Alison Simmons, we want to thank you for being here today and talking about this research. You're fulfilling a great unmet need on fistula, and we look forward to your findings in the future.
Oh, listen, thank you so much. It was really kind of you to invite me. I really enjoyed the discussion.